Good afternoon everyone. My name is Jatin Joshi and I am a researcher at Department of Biophysics, University of Delhi. Today I am excited to share with you my research on structure and functional classification of oxygenase. Did you guys know that according to the Centers for Disease Control and Prevention, over 2.8 million antibiotic-resistant infections occur in the US each year, with many involving beta-lactamase producing bacteria. To better understand and address these resistant strains, we classify the diverse types of beta-lactamase enzymes. In the next 5 minutes, I will walk you through the background of this issue, the methods we employed, our key findings, and their implications. Let's get started. So, these are my topics, and these are my education data. So, the contents of this presentation is introduction. First we talk about the introduction, then we talk about the objectives, then we talk about the methodology that we used in this research, and what we get inferred from that methodology and results. Then we talked about this, and then the conclusion part of all the research, what we got from the research, and last the references and the acknowledgements. So first, the introduction. Beta-lactamase are a superfamily of enzymes that are produced by bacterial pathogens to hydrolyze the beta-lactams. Antibiotics. Beta-lactamase are one of the major cause of antimicrobial resistance. Generally, beta-lactamase are divided into 4 classes: A, B, C, and D. These classification is also known as Ambler's classification. Among the four A, B, C, D, the D are the most diverse beta-lactamase enzymes. The OXA belongs to class D beta-lactamase and is highly diverse also at both sequence and functional level. OXA class D enzymes are the most diverse enzymes known as beta-lactamase, and these are often resistant to beta-lactam inhibitors, because of which it becomes the subject of our research. So what is the objective we try to find from it? Classification of OXA. The first objective is the classification of OXA beta-lactamase on the basis of the conserved motifs, which are specific to particular subfamily. So what are motifs? So motifs are the conserved sequences or regions in a particular peptide or protein sequence that are specific to that sequence. Next one is to identify or develop a novel antibiotics which can inhibit the class D OXA beta-lactamase. So we also try to do a hydrolyzing approach by which we try to develop or identify a novel antibiotic that can inhibit these beta-lactamase enzymes, and also we try to classify them on the basis of these actions of the hydrolyzing approach. So the first is the methodology. So as we go, the methodology, the data first we got from the BLDB. BLDB is the data source. BLDB stands for beta lactamase database. Generally, BLDB contains all the information of the knowledge in the internet about the beta lactamase enzymes. And here we have got to know that beta lactamase are classified into 4 classes: A, B, C, D. Then from there we have retrieved the data from the class D OXA enzymes. We retrieve the sequences from there on the basis of the subfam. In their class D, there is a subfam. There is a classification that there are some enzymes which are called OXA class. In OXA class, there are various subfamilies, and that subfamilies we retrieve the sequences. First of all, we remove the sequences whose are only those sequences which are from subfamily, and these sequence there are only one sequence from a particular subfamily, so we remove those. And after that we take all the sequences and uses the meme suite. Meme is also a tool that used for identifying the motifs of the sequences. As we finding the motifs, we also face that some of the sequences have active sites are present on those sequences. Those active sites are known by us by reading some research papers. Then we remove the sequences those contains active sites, because active sites are those sites which are conserved among the subfamilies. So we remove these sequences because we want to find those motifs which are specific to that subfamily. Then after that we this is the meme result. Then after that we go for the masked is also the part of the meme suite. It cross validates the data of the meme result. In masked we give all the leftover the sequences that are specific to a subfamily, and to rechecking it from the all of the sequences that we retrieved from the BLDB, then we got the result. And after that result we identify some of the subfamilies, some of the motifs which are particular specific to those subfamilies. Next, our next approach is the hydrolyzing the antibiotics based approach. The previous approach is our motif based approach, and now this approach is the hydrolyzing the antibiotic based approach. Same here, we retrieve our data from the BLDB, and from there we have taken the sequences of the class D OXA and retrieve all sequences. Then in OXA class there are various subfamilies. In these subfamilies, some of the subfamilies sequence have structure known, but some of the subfamilies have some of the sequence of the subfamily doesn't have structure. So first of all we retrieve those sequences which doesn't have structure, and we done a multiple sequence alignment with the help of EBI muscle tool. From the multiple sequence alignment, we try to identify that these sequences are conserved among the subfamilies. So that's we have to done with the help of EBI muscle tool. Then after the sequences, when we found that these are the conserved sequences among the subfamilies that, and then we use AlphaFold. AlphaFold is a tool which are developed by Google DeepMind. From AlphaFold, we predicted the structure of these sequences. After predicting the structure, we try to dog them with the ligands. The ligands are the anti-beta-lactam antibiotics which are well known around the world. The beta-lactam antibiotics such as sulbactam, tazobactam, amoxicillin, etc. are using as the ligands for these protein for docking. We use antibiotics because these sequences whose structure we determined with the help of AlphaFold, we try to find the inhibitors of them also, to know the antibiotics which inhibit these OXA proteins. We use beta lactam antibiotics, and we try to inhibit them, and then we do molecular docking. For as a machine learning approach, we also do Tanimoto similarity matrix. Tanimoto similarity matrix, in that similarity matrix we got the score between 0 to 1. The more the similar the sequence, more the value is nearer to 1. As Tanimoto similarity matrix, we use the antibiotics as the input and try to generate a matrix. After generating the matrix, we compare the Tanimoto similarity matrix with the binding affinity score which we got from the docking of beta lactam antibiotics and the OXA class sequence structures. And after that, we retrieve some of the antibiotics that inhibiting the OXA class of beta lactamase. So what we inferred from both of the approaches, the motif based approach and the hydrolyzing approach. So in the first inference, we got that from the motif based approach, we have found motifs which are specific to the subfamilies, such as this one, CQHIHTPVSF. This is specific to the OXA 214 sequence, and similarly HAQAS WNDS, which is specific to OXA 50. Similarly, we found many motifs that are specific to their subfamilies. Here, for checking the conservation, we also do validating these motifs, we use a web logo tool. Web logo generally a tool which is used to knowing the conserved region among the sequences. So if the result of our data is if web result shows some result, it means the sequences are not specific. So here web logo doesn't show any result, it means that our motifs are specific to that subfamilies. After that, from the hydrolyzing approach, we found some antibiotics which are inhibiting the beta lactamase enzymes of OXAs. Here we find that these antibiotics are inhibiting the beta lactamase. The last is the conclusion part. So what we conclude from our result? So as we know, antimicrobial resistance is a major public health concern around the world. The major reason of this is the evolution of the beta-lactamase. With the development of new class of antibiotics, bacteria are also evolving to counter these antibiotics by forming a diverse range of beta-lactamase enzymes. Categorization of these sequences into subfamilies is broadly consistent with the conventionally accepted subfamily hierarchy, although a few of the subfamilies and a few sequences within some subfamilies show inconsistency with the current subfamily classification and might reflect need for update this scheme. That's why, thus, we have identified motifs which are unique to specific family that will help us to classify them easily. We also identify some novel antibiotics that will help us to inhibit these beta-lactamase and also help in the classification of their mode of action. So at last, these are the references from where I have collected my data or the knowledge about this research. Next, I like to acknowledge my research supervisor, Professor Manish Kumar, to giving me this opportunity to work on this project. Next, I will thanks to my head of department, Professor Manisha Goyal, and then my colleagues Govinda Rai Dabaru, Akriti Jain, Somil Sharma, and Dr. Somendra Nambram for helping me doing this research. At last, all the faculty of the staff members of the Department of Biophysics, University of Delhi, and my family and friends who helps me and guide me through tough times for this tough times. Thank you.